Hi, and welcome to my presentation on my research revolving the extraction of functional insights from loss-of-function screens using deep link prediction. I'm Pieter-Paul Strybol, a PhD researcher from the University of Ghent, doing collaborative research with a biotechnology company in Belgium called Galapagos. Loss-of-function screens are experiments in which gene function is silenced using RNAi machinery or CRISPR-Cas in a certain cell line. These experiments are often used in drug target discovery to identify potential gene targets that, when silenced, revert the disease phenotype back to healthy. However, this technology comes with its own set of difficulties, one of which is the occurrence of off-target effects, where a certain construct designed to silence gene A actually silences gene B, leading to potential false positive hits. Not only that, but screening data is often very noisy, leading to the requirement of setting a very stringent threshold on the screening scores, leading to many missed potential targets. To alleviate the noisiness of loss-of-function screens and augment the final hit list, we propose a strategy based on data integration and network representation learning. Our hypothesis is that by analyzing the screening data in light of their molecular interactions, we'll identify more suitable drug targets that eventually end up in clinic. We took the screening scores from the DepMap consortium, who screened over 700 different cancer cell lines using either RNAi or CRISPR. The first step in our pipeline is to binarize the screening matrix and identify true dependencies. These are genes that show a significant association with the measured phenotype. The measured phenotype in DepMap was cell line mortality. We can then convert this adjacency-like matrix to a network where we can connect cell lines to genes if a gene is a positive dependency in a certain cell line. In this case, for example, if we knock down gene 1, we will see a very high cell line mortality in cell line B. We can then overlay this network with a gene-gene interaction network, such as the one from StringDB, for example. And what we are left with is a heterogeneous graph in which we have two node types, cell lines and genes, and two edge types, gene-gene interactions and associations between genes and cell lines. We have developed a novel network representation learning model based on a deep learning architecture that is able to predict the likelihood between any two nodes in the network. This means that we can directly measure the likelihood of any gene being a dependency in any of the screen cell lines. It works by showing the model a node pair as an input, after which our model will construct numerical vector representations for each of these nodes and assess the distance of these vectors in the embedding space. The closer the vectors are together, the more likely their interaction will be, and vice versa. The further these vectors are apart, the less likely their interaction will be. An example visualized in this slide is we give cell line B and gene 5 as an input to our model, and it predicts their interaction with a probability of 81%. This is very high and likely due to the fact that gene 1 is involved in a molecular interaction with gene 5, but is also a dependency itself in cell line B. Now going to the results, we first conducted an extensive benchmark against state-of-the-art link prediction methods and heuristic to assess our ability to correctly predict cancer dependencies in 7 different cancer types, as well as a pan-cancer setting. We did this by using either RNAi screening scores, which is the heat map on the left, or based on CRISPR screening scores on the right. We can confidently say that DLP and DLP deep walk are the best performant methods among the ones that we have tested. Another interesting observation is that the performance of almost all methods increased when trained on CRISPR screening scores, hinting towards the fact that CRISPR screens are in fact less noisy as compared to RNAi-based screens. This is also confirmed in literature. Now that we have established that we can correctly retrieve dependencies using link prediction, we can look at how many drug targets are retrieved by each method. It's important to make the distinction that not every dependency is a suitable drug target, not only because of its drugability, but also because of how it interacts with others. And this is why we believe it merits looking or analyzing screening data from a pathway-centric point of view. Here on the y-axis, we see the performance of each method in correctly prioritizing known drug targets. And we can again confidently say that DLP DeepWalk identifies or prioritizes known drug targets significantly higher up in the ranking as compared to the baseline of just using RNAi scores, which is the black dashed line. This causes valid or potential drug targets to be picked up more easily. Another observation from this plot is that there is indeed a correlation between correctly retrieving cancer dependencies and highly prioritizing known suitable drug targets. Finally, we attempted to reconstruct the mode of action of perturbing a gene either through knockout or knockdown or by attacking it with a drug. In this case, we constructed a subnetwork around a known drug target, GIF11, by combining their gene-gene and gene-cell line interaction probabilities. Through gene-set enrichment analysis, we confirmed the role of the subnetwork in spindle dynamics and cell cycle, which is in line with the described mode of action of GIF11 inhibitors. Furthermore, we also identified many other drug targets in this network, for example, AORGB. And AORGB is actually known to have a synergistic effect together when used in combination with GIF11 inhibitors, showing the value of looking at hits or drug targets from a pathway point of view. I thank you very much for listening and kindly refer to our publication for more detailed information.